Hello guys, welcome to my channel BioBCC Basic Concepts Cleared. Today we are going to deal with one of the most important topics in molecular biology, which is PCR, Polymerase Chain Reaction. So let's start. Polymerase chain reaction is done in a device known as thermal cycler. Why cycler? Because we have to conduct cycles over and over of the same process. And the basic three steps in this process are: first is denaturation. As we all know, most of the 90% of the eukaryotic organisms, higher eukaryotes, have double-stranded DNA, which has hydrogen bonds between it, and it is double-stranded. It is complementary. And for the denaturation, we use higher temperature so that the bonds between the two complementary strands degrade and then form a single-stranded DNA. The second step here comes is annealing. Annealing means attachment. What are we attaching? We are attaching a primer. Primer is basically a single-stranded RNA, a short sequence of RNA, which is known to us. Means the sequence of the primer is known to us, and we attach it by adding to the culture mix at 55 degrees Celsius to 65 degrees Celsius. This is 6, not 5. So as you can see, for denaturation there is a higher temperature, 94 degrees Celsius, whereas for annealing of primer the temperature is lowered. Why so? Because we have to attach the primer, right? We don't have to break the hydrogen bonds between the primer and the complementary sequence. At the third step, extension happens, which is again a rise in temperature at 72 degrees Celsius. Now 72 degrees Celsius is also a high temperature, we can say. So the normal polymerase is inactivated. The protein folding of the normal polymerase which we have is inactivated at such high temperature. So we need a special type of polymerase. This is the polymerase. It is known as Taq polymerase. Why Taq? It just means Thermus aquaticus polymerase. It is derived from a microorganism which lives in a very high temperature in hot springs, known as Thermus aquaticus. The polymerase from this organism is isolated, and then we use it for our PCR. So after this whole cycle, after this one complete cycle, first we had a template DNA which was two-stranded. Okay. After denaturation, annealing and extension, now we have four strands of DNA, four single strands of DNA. Okay. This cycle is repeated on and on to get a very higher amount of same DNA strands. As the cycle proceeds, we amplify the DNA. These are the components required for the PCR. First is the DNA sample. The DNA sample. Second is the primers. Third are the nucleotides. A, T, G, C are the four nucleotides which are used. So they are totally, we can say, they are usually known as DNTPs. And we have to provide these because the Taq polymerase has to add something to the single-stranded DNA, and this is what is added, the DNTPs which we provide. Now there is a mix buffer to keep the DNA stable, and this all reaction happens in a PCR tube, or also known as microfuge tubes. And all process this all process happens in the thermal cycler. That's it for today, guys, and please like, share and subscribe to the channel Bio BBC Basic Concepts Cleared, and I hope you have a great day. Thank you.